I'll start with a brief introduction. I mean, I don't need to introduce the importance of research on aging and tackling aging and develop interventions for aging. We've heard quite a few talks on this already. And we've also heard that there's reason to be optimistic in the sense that we can now manipulate the process of aging. As we've discovered in the past 20, 30 years, aging is a plastic process, particularly at the genetic level. It's very impressive how much we can manipulate aging. So they started with, well, just a reminder with age 1, which was identified 30 years ago by Tom Johnson and colleagues in C. elegans. And now we know that the process of aging can be accelerated and can be retarded, as we've heard already at this conference. And this is really, I think, a major breakthrough in the field. So as some of you know, one of the projects that, well, I actually started when I was a PhD student here in Belgium many years ago, is a collection of databases that includes the GeneAge database of aging-related genes. And I'll just focus here on that we now know of over 2,000 genes in model systems, that is, mice, flies, worms, and yeast, that when genetically manipulated, overexpression, disruption, mutation, and so on, significantly impact on aging. So again, this is a huge, I think, reflection of how much we have advanced in our understanding of the genetics of aging in model organisms in the past 10, 20, 30 years. Just as a side note, one of the latest databases that we've developed is a database of genes associated with cell senescence. So we call it Cell Age Database. Since we heard quite a few talks, including the previous speaker, Thomas, on cell senescence, I thought I would just mention this. So we have about a little over 250 genes associated with cell senescence. So again, when you genetically manipulate them, in human cells, it has a significant impact on the senescent phenotype, and this is available online. So it's still not finished, but we have a beta version available now, and I hope that will be also useful for people who are working on cell senescence and on aging. So that's just a side note. But the point is that there's a lot of genes that impact on aging in model systems, but of course it's not straightforward. We cannot just change genes in human beings. So what a lot of approaches are focusing on, and is now developing pharmacological approaches that allows us to manipulate aging. And again, we've heard quite a few of this in this conference. I mean, rapamycin, well, Thomas mentioned metformin, resveratrol, etc. So there's a variety of pharmacological interventions in aging that may allow us to intervene in the human aging process. So again, we've been cataloging this as well. So this was something we published last year, so I won't spend too much time on it. But we also have a database of aging-related drugs called DrugAge. And just to give an overview of this, it's based on over 300 papers and over 20 model systems. But my point is that it includes over 400 drugs or compounds that significantly extend lifespan in at least one model system. So again, this is a reflection of the advances that we as a field of biogerontology have achieved in identifying this, well, some people call geroprotectors, geroprotectant compounds and molecules, which really opens the door for potential human application. So this is, I mean, our database is only based on model systems, only based on model organisms so far. The other advance that we have seen, and that's something that our lab has been focusing a lot, is in big data approaches and computational biology, in particular driven by technological advances that allows us to sequence DNA cheaper and quicker than ever before. So you see machines like this one. I mean, this is a photo of me pretending I work with that machine. But what I do know is that the cost of sequencing, so this is a log scale, the cost of sequencing per genome or per DNA has been decreasing very rapidly in recent years. So it's much, much easier and cheaper now to sequence your own DNA and to, of course, do high throughput experiments in the lab. So really the shift has been from generating data, which is much easier and cheaper than ever before, to analyzing that data. And so I'll quickly give you a couple of examples of approaches we've been employing. In particular, well, I'll only focus on pharmacological approaches. So one project we've been involving is in terms of repurposing drugs for identifying new geroprotectors or identifying new life-extending compounds. So again, we published this 2 years ago, so I won't go into methodological details, but I mean, the paper has been published, and feel free to ask questions or talk to me afterwards. But essentially, we look at signatures of longevity, gene expression signatures of longevity from high throughput studies. Specifically, or in what we published so far, we looked at caloric restriction. And then you can obtain gene expression signatures from compounds, from drugs, from public repositories. And so we looked for drugs that induce a gene expression profile, a gene expression signature that overlaps with that of longevity. Well, as I said, we use, for this particular project, we use caloric restriction. When we have some ongoing work where we use other gene expression signatures, including cell senescence, but for this we use specifically caloric restriction. And then we found, you know, we do statistics on it, and I won't go into details on that, but we identify and rank compounds that overlap at a statistically significant level with that of longevity signatures. And we decided to test this in worms because it's fairly cheap and quick to do these experiments. And of the 5 drugs we tested in worms, 4 of them extended lifespan. I mean, that includes rapamycin, which was a sort of positive control. So what this shows is, because we can employ this, we call this network pharmacology approach, to identify and rank compounds that may have geroprotector or life-extending properties. And I said I won't go into a lot of details, but one of the, well, I thought the most interesting compound we'd identified was allantoin, which is a marker of oxidative stress, but as far as I know, we were the first to show it extends lifespan in any model systems. But because it's a marker of oxidative stress, it's quite an intriguing one. And I mean, there's some results now suggesting that it acts also synergistically with rapamycin. This is from a group in Singapore, Jan Gruber's lab in Singapore. And we're trying to figure out the mechanism for allantoin and also other similar compounds in the same that work in similar ways that may have applications. So allantoin is used already. So if you buy some skin aging creams, some of them contain allantoin. So it is relatively safe. And so we think that this way of repurposing drugs for longevity and as geroprotectors, because we have informations from the drugs already of their usages and potential potential side effects, is one promising approach is to identify geroprotectors. Then the other thing we did, and again, we published this last year, so I won't go into a lot of detail. This is, I'd say, a complementary approach. We employed a machine learning approach for predicting life-extending compounds. So the way we work is we build predictive models based on the properties of these chemical entities. So that involves chemical features and also biological features of the targets of the chemical. So you have a drug and you know what target it has, and then you can use the biological features of its target. And then we build predictive models of which chemical compounds extend lifespan and which don't. And the best model we employed, it employs a combination of biological and chemical features. And then we can apply that model to other compounds and try to predict new life-extending compounds. And most of the hints were related to chemical compounds that target mitochondria, quite a few cancer-related drugs, and also inflammation, so processes that you would expect. But again, this was done in collaborations with Alex Freitas in Kent, and it was funded by the Leverhulme Trust. This is another complementary approach in terms of drug repurposing. We employed a network pharmacology, which I mentioned earlier, and this is a machine learning based on the chemical and biological features of chemical entities. So we, based on all of these developments, it's not surprising that we have this growing anti-aging biotech sector, and we've heard quite a bit from this in this conference already. I mean, Calico is probably the most famous from Google, Unity as well, which I think have been mentioned, working on Senolytics and with funds from Jeff Bezos from Amazon. And I mean, this is sort of my disclaimer as well for the companies that I do some work with. Now, so there is a growing biotech industry focused on anti-aging, on the anti-aging sector. But if I can, play devil's advocate for a second, there are some issues in translating findings to the clinic and in developing interventions in this context that I just wanted to point out. So as I'm sure you're aware, most companies, only a very small percent of companies, biotech companies become profitable, most of them fail, and likewise, only a very small number and very small percentage of compounds actually obtains regulatory approval. So most companies and most compounds fail. And so that's for the whole biotech industry. And in our own field of aging, we have the problem that there are long validation times because we don't yet have precise endpoints or established endpoints for clinical trials. As it's been mentioned before, metformin is undergoing a clinical trial, and hopefully that will allow a proof of concept for how to develop clinical trials in the context of aging. So those are some of the problems. The other problems, well, there's a couple of other problems I'll point out. And the reason I do this is that, obviously in the end, I would like some of this to succeed and try to address as many of these problems as possible so that we actually, what I really like would be to have some molecule and some compound or some intervention that really allows people to live longer by retarding the process of aging. So a couple of other problems. So let me tell you a story. So again, this started in Belgium when I was a PhD student. So I was doing some statistical analysis of mortality. And again, without going into a lot of details, what this shows is, so this is hazard rates or mortality, and this is age for mice. And these are controls, and these are mice-fed fish oil. So what this shows is that in this strain of mice, fish oil retards the demographic pace of aging, which I thought was quite interesting. And so we published this many years ago, but that led to me putting in a proposal to the interventions testing program of the NIA and the NHS, of the National Institute of Aging for testing fish oil, which was published 2 years ago. And long story short, when you feed fish oil in their program, it doesn't extend lifespan. Okay, so this is data for males, but the data for females shows exactly the same, no trend or no effects on lifespan. Now you might have noticed this is a relatively short-lived strain. They only live about 1 year. So this is a particularly short-lived strain. An intervention testing program, they employ an heterozygous, they combine a cross. There is not just a single strain. And as I'm sure you're aware as well, or some of you are aware, caloric restriction does have different effects depending on the mouse genetic background. So the point with this is that we have a problem in terms of a lot of experiments done in mice in particular strains that may not be applicable to other strains. And that is a problem, particularly when you think about human beings, which of course are much more heterogeneous genetically and environmentally. So that's one problem, is strain-specific effects. The other problem is differences between species, which was something that's come up already. So let me show you. So this is from our drug age database. And this is, what you see here is for different animals, so worms, flies, and mice. You see the kind of lifespan effects, life-extending effects from the compounds that we have in the database. And you see quite a wide range in worms, slightly narrower in flies, and a much narrower, much smaller effect sizes in mice. So the point is that you see this as you increase complexity and more similar animals to human beings, you see smaller effect sizes. And so we still have this problem of, okay, so what happens in humans, which was my questions to Thomas. We still don't understand human aging particularly well. I would say we still have a poor understanding of biological aging. We have a lot of data, for example, for senescence in mice, but we still don't understand well whether that is the case. We have relatively little data on most mechanisms of aging for human beings. At the genetic level as well, although we've had great advances in our understanding of aging genetics or the genetics of aging in model systems, we still have a poor understanding of the genetic basis of human longevity. And so the big question is, okay, can these interventions work in humans? And that still remains a big question mark. And even if they do work, how much of an effect will they be? Will it be better than just going to the gym? Because we are aware of interventions that are healthy for us. So will this pharmacological interventions in particular, will they be better than just regular exercise? Now, one of the things, before I finish, I will point out that one of the things we are trying to do is to incorporate more human data. So try to bring into our predictive models, in our computational approaches, human data that I wouldn't say will tell us the answers for sure, but may allows us to inform into interventions on aging or genetics of aging. So, for example, we have databases focused on human genetic variants associated with longevity. We're also employing data from the Framingham Heart Study and UK Biobank, which Thomas mentioned as well, which has over 500,000 individuals trying to discover genes and associated with aging phenotypes. And also looking at patients. For example, we're working with Twins UK in King's College to look at patients that are taking particular drugs and see if that affects their epigenetic clock, for example. So that is the type of information from human data that we're trying to include in our analysis and in our models to try to predict as best as we can, or try to guess as best as we can whether particular interventions or genes will be relevant for human aging. So with that, I will summarize. So I've told you that we know a lot about aging manipulations in animal models. So we know of over 2,000 genes associated with aging in model systems, which again is it's remarkable. It's really a huge breakthrough. It's very exciting. And we know of over 400 drugs that are associated or that significantly increase lifespan. So that is very remarkable how much we have advanced in model organisms. I told you of our recent cell age database of genes associated with cell senescence. I told you of our computational approaches to prioritize drugs, including network pharmacology approaches, as well as machine learning approaches to identify and rank and prioritize compounds that can act as geroprotectors, which we then tested in worms, and we're still testing. But I hopefully persuade you as well that there are huge risks in human translation and there's huge unknowns. And so, although there is a lot of excitement in the field, most companies, most of these biotech companies will fail, most clinical trials will fail. And ideally, we want to have human data to inform and to better select our candidates for human translation, and that's something we are trying to do, trying to integrate human data into our models. And so with that, let me just thank this. So this is our various lab members, and I mean Daniel has been working on our various databases, including Cell Age, as well as Roberto, and Daniel Palmer as well, the various gene expression analysis that I've mentioned. So these are the people from our lab, and these are our very helpful and gracious funding sources. And thank you very much for your time and attention.